Hi, everybody. Thanks for waiting while people were joining. We've got lots of people participating, which is great to see. Um, so first up, I'd just like to, on behalf of the ARC Centre, acknowledge and pay our respects to the original owners and custodians of the lands, the many lands on which we meet today, and recognise their valuable contribution to Australian and global society. So my name is Tim Brodrup. I'm one of the chief investigators at the Centre of Excellence for Plant Success, beautifully named ARC Centre of Excellence. And it's my great pleasure today to introduce Professor Missy Holbrook, who's the Bullard Professor of Forestry at Harvard University. It's a particular pleasure because I did my first postdoc with Missy many years ago, and it was a fabulous experience that went for many years, and you know, we had a fantastic working relationship, and Missy's become a good friend of mine. But Missy's been a tremendous force in hydraulics for many years. She's one of the most outstanding sort of experimental biologists that we have in water relations, and she's just got this, you know, she's got a very rare understanding of plant physiology and plant biophysics, which has really allowed her to make fabulous contributions to these diverse disciplines that hydraulics seems to now be involved in, which, you know, goes from ecology to molecular biology and evolution. So yeah, it's a great pleasure to be able to introduce Missy today, and if you've got questions while Missy's talking, you can put them in the chat, or if it's a really burning question, Missy said she's happy to answer questions if people interject. So that's also a possibility. So I should hand over to Missy now, and we'll hear about what she's been up to.
Thank you so much, Tim, and thanks everyone. And yes, as best we can on Zoom, I like to make it informal and interactive. So very fun to be here, and thank you. So I'm gonna let's see, I'm gonna share screen. It'll take me 1 second to do that. I'm gonna have to use glasses now. Sorry about that. And make sure then. Okay, so I want to frame my talk in the sort of the key issue in biology are trade-offs. So here is a male peacock, and these feathers, this plumage, faces, you know, it's great for attracting and mates, really bad for evading predators. And in plant biology, you know, a key trade-off that it's very fundamental to plants is the trade-off between speed and specificity of rubisco. That's a really, you know, fundamental molecular issue that has a lot of consequences for across every scale of photosynthetic organisms. Today, I want to talk about a different trade-off, a different sort of design constraint faced by the xylem, and that is being strong enough to withstand the negative pressures that are the driving gradient for water movement through the xylem, and the need to move. So by having strong and stiff walls allows these water-filled spaces to persist under, you know, really large negative pressures, so inward on the wall, while at the same time allowing water to pass through these walls. So you see here in the axial xylem and a stem of an ulmus that let me pop on a laser pointer. That we have pitting, so we have regions of the wall that are weak but permeable. Now, I don't want to stay in the transport xylem, but I want to move up to the end of the transpiration stream where we're in the leaves now, and the water is going to must leave the xylem and move out to irrigate the photosynthetic cells, and eventually to replace water that evaporates and diffuses out through the stomata. And here we see not only the largest tensions that occur along the gradient, but also these dual features of having to be reinforced to allow negative pressures to occur, but to be permeable to allow the water to leave. And I've been long interested in what happens at the end of the transpiration stream where these mechanical and design hydraulic constraints come together. And so I was very excited to see a paper, and this is right before I was coming for my very first visit to Tim's lab, or to visit Tim in Tasmania. Well, so sorry, I'll talk about that paper in a second. Sorry. So when we think about these conduits, and what we'll see is that they actually do collapse. So now they're pushing that trade-off between strength and permeability. And if they do, you can get these conduits to actually collapse, like a drinking straw if you suck too hard on it. And I want to ask the question, is there a functional role for minor vein collapse? And I've put on this picture here of Oliver Wendell Holmes, was an American poet and philosopher. He was also a dean here at Harvard, and he wrote what is a famous poem for people who work in biomechanics called The Wonderful One-Horse Shay, a shay being a carriage, in which every piece of this carriage is designed, is equally strong so that the whole thing wears out at once. No part is overbuilt with respect to another. So the question then is, if these minor veins, the end of the transpiration stream does collapse, is that just a reflection of not over investing, or is this something that is used, has an adaptive contribution to plant hydraulics? Okay, and there's the paper I was jumping ahead to, because I had long wondered whether this might happen in plants, and I always thought it would be unstable and you could never observe it. And so in 2004, Hervé Cochard and colleagues published this paper in Plant Physiology on pine needles. And so these I'm just showing a few of their images of Pinus sembra, using cryo-SEM at a variety of, increasingly negative water potentials. And these arrows show that we're starting to see buckling and deformation of the tracheids and the xylem here at -5 megapascals. This xylem is nearly unrecognizable. It is so crushed together. And with a little additional tension, a different sample, because these are all frozen samples. That's why you're able to see the persistence of these deformed state. These are now air-filled, and that the water has lost its metastability, its negative pressure, and the walls have relaxed back to their original conformation. Here in a different species in Pinus nigra, here's the xylem. This is using a fluorescence technique and hydrated state. The tracheids in cross-section are nice and boxy around. Here's some rays going through, and here's at -3.5 megapascals showing this crushing. And this is falls a little bit in the line of, here is so for Pinus nigra, here is the transpiration rate, so the stomata shutting, so that's declining. And here is the predicted loss of conductivity due to this deformation, this crushing of the tracheids that Hervé and colleagues have calculated here. Note that this is happening at a more hydrated state. So this is the minimum water potential that these needles experience in megapascals, so going from fully hydrated to more negative here. And all of this is happening, stomata are shutting, tracheids are collapsing or buckling before cavitation is occurring, either in the stem or in the needles. Now this is, so when Tim and I saw this paper, we were excited about this. And as I said, this is when I came to visit for the first time. And this was a real inspiration for a study that we did together on Podocarpus grayi. So I'm not going to show too much of this, but it's a beautiful plant from far north Queensland. In fact, don't know the entire distribution, because we worked on several plants that were in the glass house at the University of Tasmania. But what's amazing about this plant, so it's a conifer, so it has a single vein, and like all conifers, it has this horizontally oriented transfusion tissue. But to have these very broad veins in the Podocarpus, it has not only the normal transfusion tissue, so here's the axial xylem, here are these sort of boxy transfusion tracheids, and then it has these long tubular, what are called accessory transfusion trachea, that basically make it a sort of a continuous plane of xylem that goes out and allows these conifers to have these very broad lamina. Okay, so here. So it has the perfect orientation, and these are very large tubes to look for deformation. So we use the epifluorescence with a little, literally a Dixie cup or that's what we call it in the US, a styrofoam cup of liquid nitrogen under a fluorescent scope, and we were able to see, so now here's a cross section of the leaf with the palisade at the top and the lower epidermis here. Here are the accessory transfusion tissue. These lignified cells are showing blue in their autofluorescence here, and here is a not stressed, so -1 megapascal, and these tubes are very round in cross section. So here's the slice, and then at a more lower, more negative water potential, you can see that these tubes have completely collapsed. So this was amazing, I have to say, and there were subsequent measurements made. I have to acknowledge, I would say, I was going to say mostly by Tim, but I'm going to say they're all made by Tim, because honestly, I don't think I stayed long enough to do this. But what it shows, what's really interesting here, so let me walk you through this figure. We've got leaf hydraulic conductance in both the A and the B panel, and xylem water potential here is going from hydrated to more stressed over here. And the conductive system of the xylem is in these black diamonds, and it's going down. Okay, so it has this, there's a loss function, and it's corresponding with where the stomata shut. So this is what this line here indicates. Now, at the same time that that's happening, so not afterwards, but coincident with that, we see that the loss of these, I'm going to try and move this so I can see, I can't do that. Okay, the tracheids are collapsing. So now it's not like the tracheids are collapsing after the stomata shut, which would sort of indicate, well, don't be overbuilt, but they're happening at the same time, leading to this idea that maybe it's this sort of choking back of the hydraulics of these tubes that then means the stomata run out of water and they shut. Notice that all of this is happening prior to the water potentials that cause cavitation, which are up here in this solid line here, and these are more negative than we're having for the collapse. So they're happening subsequent to stomatal closure and the accessory transfusion tissues collapsing. So this led us to hypothesize that this collapse, it coincides with the decline in leaf hydraulic conductance and stomatal closure, and that it might be have a causal relationship with stomatal closure, thereby protecting the leaf from reaching water potentials that could cause cavitation. We went on then a number of years later to do some additional work in a conifer here in the United States, a Taxus baccata, which is, I think actually is a European species grown horticulturally here. And it's actually the first species in which transfusion tracheid tissue was described. So here's a cross-section of the vascular bundle in these needles. So the phloem's on the bottom. These needles live for a couple of years. So you get a couple of years of phloem. You've got xylem up here, just this little bit up here. And then you've got these boxy transfusion tracheids. And these are really tracheids. They're lignified, weakly lignified. They undergo a programmed cell death. They have no cell contents at maturity. And they are where water enters the vascular bundle. And there's been beautiful work in the last couple of years done by Thomas Bohr and Alexander Schultz and their colleagues in Copenhagen on looking at how these sort of entry points of water on these sort of on the polar sides of the xylem in these linear leaves is critical for helping water come in into certain parts or connecting to the xylem in certain parts, and ditto for the phloem. So there's been really nice work on this sort of the function of this stuff. We got interested in these transfusion tracheids because they're so big and boxy, and it looked like if anything was going to fall apart and collapse and be mechanically unstable, it would be these guys. So here's the picture in the epifluorescence. It's a frozen section. Here is a cryo-SEM. The X represents the xylem. You can see these tracheids here, and then the transfusion tracheids are these larger, more irregular shaped cells here, and then here's a longitudinal section showing the axial xylem and these very boxy transfusion tracheids on the side. There's no accessory transfusion tissue in this species. So we have studied it using this frozen epifluorescence technique, where we can see what was nice about this, we could see the collapsed transfusion tissue. So here at minus 3.4 megapascals, you can see these more open cells are now very kind of crushed together. And if you take the same sample and you rehydrate it, you can then freeze it again and look at it, put it back in the liquid nitrogen, and you can see, in fact, that the tissue has rebounded. Okay, so when you relieve the stress, it opens back up. So, and then we were able to, we can also do this using the cryo-SEM. So here is the boxy xylem, and here are the crushed transfusion tracheids at -2.5 and -4.4 megapascals. It's very hard to see because the stuff is just so completely squished, but after rehydrating leaves, after drying them out and then rehydrating them, you could see the transfusion tissue regains its shape, and it's still filled with water. So this was one of the big surprises for me. I always thought that these cells, when they became mechanically perturbed, would immediately cavitate, and you would never really be able to see it. It would be too fleeting. But it turns out that these are actually fairly robust, the water under tension, to both the deformation. It stays ice-filled here. So it was water-filled when it was frozen to make the image. And these guys were able to rehydrate without cavitating. So they were dried, rehydrated, then frozen. So one of the things we did is we made leaf hydraulic conductivity measurements. We used the rehydration kinetics method. So that's the K-leaf. It's a measure of the ease with which water flows into the leaf. So normalized by the leaf surface area, so millimoles per meter squared per second per megapascal of driving force. And the K-leaf measurements are in the gray, and you'll see as the water potential goes from more hydrated to more negative, the hydraulic properties of the leaf decline, and that's these gray line here. And we overlaid that with a plot of the transfusion tracheid area. So we just basically took those epifluorescence images and we outlined the area of transfusion tracheids, both when they were in the stress state, so however dry we were, and then after we rehydrated up to fully hydrated to get a percent. So when it's hydrated, it starts at 100%, and then it declines. And what we found was they overlaid, perfectly in terms of when this decrease in the relative transfusion tracheid area occurred, and when in the water potential, with when we saw this loss in the hydraulic conductance of the leaf. Again, it's very suggestive that this was an important driver of this decline in K-leaf. We followed this up. We said, we wanted to know, that's very suggestive, but is cavitation also contributing? We knew something about cavitation in the stems of Taxus baccata, but less in the leaves. And what we did in this case was we took leaves. So the black points are the ones you just saw. That's the native K-leaf, as we dried leaves down to different water potentials and then measured their hydraulic properties, how their ability to take up water at those water potentials. And then we took leaves that we dried down to a variety of water potentials, and then we rehydrated them up to 1 megapascal and measured their hydraulic properties there. We wanted to see how much of this loss was reversed when we relax it to -1. And so those are the points in white. You can see they start all, had to be dried below -1 before we could hydrate them back to -1. And so that was what we call, that's the irreversible part. It's the part of this decline that was not reversed when we hydrated the leaves back to -1 megapascal. And the difference then is this reversible part, which we feel, since cavitation is not reversible in these leaves, that most of the decline here is attributable to the collapse of the transfusion tracheids and not to cavitation. There is still some decline, there is some cavitation, there is some overlap out in this part of the water potential domain that is due to cavitation. But by that point, the collapse of the transfusion tracheids have already choked down the hydraulic conductivity so much that this is probably not an important part of controlling what's going on with the stomata. And you see I have sort of midday leaf water potential plotted here, and the point of stomatal closure here. So coinciding with the, preceding when cavitation occurs and coinciding when collapse occurs. So again, it's very suggestive that in these species that collapse could be playing a role in potentiating stomatal closure to happen in advance of cavitation. So finally, I want to get to my third sort of plant example and spend most of my time here talking about Quercus rubra, the red oak, which is really a very important species in the forest here in Massachusetts. It's a real dominant part of net primary productivity. It's a deeply rooted and highly productive tree in this area, and has really nice leaves to work on. They have really robust petioles, big lamina, and they can photosynthesize and transpire at pretty good rates. It's a ring porous species, has high hydraulic conductivity. And it's a heterobaric, like most leaf orientation, which means that its minor veins form distinct areoles with bundle sheath extensions up to the upper and lower epidermis, but then it has intrusive veins, and a lot of them that go inside the areoles that have no bundle sheath epidermis, but sort of float within the mesophyll. And this really, to my eye, very pretty image from my colleague Juan Losada, the confocal, showing, so here is a vein that's bounding an areole, and here is an intrusive vein that's coming in and terminating inside the areole. These are some photosynthetic cells that are fluorescing beautifully in this image. And what I want to draw your attention to here is just that the incomplete lignification, with lignin showing here in yellow, of these tracheids in these highest order veins, and then the termination in these really kind of fun bulbous, sort of fenestrated of cells, which are, where the water is leaving now the xylem to move outward through the bundle sheath cells. You can see some of them outlined in yellow here, and into the mesophyll, and some here with the spiral. So we have a really kind of a change in morphology when we get to the end of the transpiration stream. And I'm long interested in sort of what's going on with these very distinctly shaped cells. So we did this study primarily using cryo-SEM. So here's what is for a red oak, a well-hydrated leaf. So at minus 1.4 megapascals, this would be a very happy leaf on an oak tree, with the upper epidermis has a large cells here, the palisade, and then quite a few of these sort of intrusive or floating veins that are inside of the areoles. They often branch, so you'll get a group of them together like these, and we know that these don't have bundle sheath extensions, so they're sort of sitting there in the mesophyll. We have a small spongy and some stomata down here. So very nice plump cells, and a really, like lovely look at these tracheids. And if you spend enough time looking at these, you start to understand correlate the texture of the frozen material, that helps you see that this was water when it was frozen, and this is a vacuole. This is a bundle sheath cell with some cytoplasmic contents here, and these are tracheids here, and these are all bundle sheath cells. Okay, so that's what it looks like when it's happy. And here's a close-up, and again you could see these regions where the solutes might get accumulated by the freezing process, the very thick walls. So a number of characteristics help us identify what's a tracheid, what's a bundle sheath cell, in addition to position, but primarily the texture here. In the intrusive veins, we tend not to have phloem go all the way to the end. It's just the tracheids. Okay, so here's a well-hydrated, and here's a stressed leaf. You can see it's stressed by the contortion of the mesophyll cells as the bundle sheath cells are shrinking, but we can also see it by the fact that these tracheids are so crushed. So here I'm going to outline, this is a tracheid that kind of has this kind of shape here. Here's another one here. Here's one that has kind of a kink in it. It's kind of like a buckled. And then here's one that looks pretty round. And here's one that is very, very flattened. Now this one that's round, it's possible that out of plane it is further collapsed. It's one of the limitations of these being able to make just a single section, that we can't really say for sure what the full properties of the cell are. We only get one section to sample on. Okay, so to quantify, to characterize the impact that these cell deformation might have on water transport, we just defined a hydraulically weighted index of roundness. And this is based on our area and perimeter of each conduct, because we needed a way to do more than just say, this one looks really squished. So our index was calculated. We divided the area of the conduit by the area of a circle with the equivalent perimeter. And then we squared the ratio to make it proportional to the fourth power of the radii, as in Hagen-Poiseuille equation for pipe flow. So our hydraulic index has a maximum of 1, and it decays to 0 when the tube is completely flattened. Okay, and here, so now I'm showing you a series of fine veins, just exemplars from -1 megapascal, -2.5, so that's approaching the Turger loss point in red oak. -3 is starting to be stressed, and -3.5 is the one I just showed you, much more stress. And on each of these conduits, I have put the numeric value of the hydraulic index, so you can see in the they're not perfectly round, so they don't start at 1, but they start at high values, and then they start to decrease. We see some heterogeneity in how the conduits change, and they go down to some, very low numbers, this one being 0.01. So, this was our hydraulic characterization of the impact of this cell deformation. Okay, and then we did this, and we calculated how the hydraulic index changed for all of the fine veins, and we also segregated 2 types. We have the type 1 or type I, really being these intrusive veins, the ones that have no bundle sheath extension that run out into the areoles, and then the B are what we would call the really the minor veins, because that you would see if you looked at the leaf, they're the highest order vein that has a bundle sheath extension. So they define the areoles. And we were able to see by drawing leaves to many different water potentials, going from hydrated down to -4 megapascals, how the ensemble hydraulic index of all the veins that we measured changed, and we see sort of a loss function. We could describe that with a Weibull function here. Now, you may say, why didn't you just measure the hydraulic conductivity and measure the effects of the collapse on the hydraulics? And this stumped us for a while. And the problem is, and that was not a problem with the conifers, is that when we use rehydration kinetics to make our measurements, the reversibility, the relaxation of these conduits was so fast that in the process of the measurement, we lost the impact of the collapse. In other words, the reversibility was just so reversible that we were not able to reliably detect it. So we could do that. We could do hydraulics on the petioles, which were losing conductivity just due to cavitation. There was no evidence of collapse in the petiole itself. But when we tried to measure it in the leaf using the same techniques we'd used in the conifers, we didn't see that. And subsequently, we did work with how fast the veins could pop back to their original state. And it was within the measurement time that we needed to resolve our rehydration kinetics. So what you see here, and what I've circled in red, so this is our hydraulic index for all the veins, the intrusive veins, the boundary veins, the petiole percent, the loss of conductivity, and then the K leaf, the hydraulic, our measurement of the hydraulics of the leaf. And this is the loss function using a Weibull fit, and these are all the fits. But the focus here is the xylem at 50% loss. It's like a LD50 or a P50. What is the water potential at which we have lost 50% of whatever our quantity is here? And the important takeaway here is that the most positive values occur for the hydraulic index, particularly of these intrusive veins, which make up a huge fraction of the total vein area of these leaves, and where we think the majority of the water may leave the xylem as it irrigates the mesophyll, and these are the most positive. The petiole is losing conductivity due to cavitation at more negative water potential, as is the entire leaf. When we subsequent to this, we use the technique that Tim has so beautifully developed and pioneered and invented this optical technique, we also showed that the cavitation P50 for leaf veins was similar to what we had seen for our K leaf. Sort of, these were done on a different year, so it's from the same plant. So again, in the negative range here, more negative than we're seeing the collapse. Let's see where I'm at. Okay, so now we've faced, this question, like, this is happening. Is this important? Is it valuable does it have a functional role, or is it, a case of don't overbuild? So we first did this with a model. We made a very simple sort of resistor model, RC model, because we've got a leaf capacitance, and we had a model in which the only variation in the hydraulic pathway was due to cavitation. So stems could cavitate, veins could cavitate, and there was transpiration, E total coming out. Water could come through the veins from the soil, or it come out of capacitance in the leaf from living cells. Okay. We had a second model, which is we call the cavitation and collapse, where we separate the transport through the major veins, which we did never observe collapse, and we say they can only cavitate in our model, and the minor veins, which can collapse. And so we have and again, transpiration going through. And then we looked at the behavior of these two very simple models when we challenged them by doubling transpiration and prevented the stomata from closing. Okay. I'll show you those model results in a second. Now you may say, well, that's not that, why would you, how relevant is something like that to a plant? But, red oak is a canopy species, and up in the canopy, the temperature of a leaf, and so these are measurements on red oak leaves at the Harvard Forest canopy, fluctuate over very short time steps by many, many degrees. And that means faster than the stomata can respond. And furthermore, as the leaf heats up and water starts to leave the leaf, you will get the first response will be an opening of the stomata due to the hydropassive interaction with epidermal cells. So we thought over these short time spans, if the stomata are open, there could be a leaf could be subjected to very large swings in transpiration without the time to be able to osmotically adjust guard cell turgor. So we thought that this was a reasonable, from the point of view of a leaf, not in a cuvette, but in the real world, that this might be a reasonable perturbation. So let me walk you through the model. I'm going to start on the side with the cavitation only model, which is a little more straightforward. And we're going to do at time 0. I'm going to double transpiration, not going to let the stomata shut. This is a plant without stomata. I'm going to run it for about 10 minutes, so 600 seconds. Okay. I've got 3 graphs here. I've got the water potential. I've got transpiration as a fraction of the total transpiration. Okay. And it's going to double at time 0 and then stay constant across the graph. Okay. And I've got the hydraulic conductivity as a fraction of the initial. So it's going to be what happens to it. Okay. So in our cavitation only model at time 0, the transpiration doubles. The water that is coming through the veins is here in red. This is the percent of E supplied by the xylem. And it's half because all of a sudden you've added the second half, and that's got to come initially out of capacitance. So half comes out of the leaves, but then the driving gradient, the leaf water potential starts to fall, more water flows through the xylem. But as the water potential falls, so you start to, when you reach water potentials that are damaging to the leaf, the conductivity declines and it declines monotonically. The leaf water potential continues to crash. And now the water that can come through the veins goes to 0 because they're all cavitated, and the leaf just dries out till it's crispy. The petiole water potential recovers because it's still attached to the plant and the soil, and it's now disconnected from the transpiring leaf by cavitation. So this is the kind of, hydraulic failure model, very that leads, is described as leading to, during drought, could lead to plant mortality. So let me walk you through then the parallel model with the cavitation and collapse. And the graphs are the same. And at the time we at time 0, we double the transpiration. The transpiration is going through the veins, but it doesn't go down because the major veins do not collapse. Okay, so so some water continues to flow through the veins, but and some comes out of capacitance. So what happens is, as the leaf water potential falls, is it causes the minor veins to collapse. They choke down on the flow, then it keeps the water potential of the xylem of the vein, so that's here in red, from falling to the values that would cause cavitation. So the K of the vein stays, it goes down some. There's a little decline, but not a lot. So the leaf as a whole, its K declines, but it's done by this reversible collapse and not by cavitation. And so this is in a model space evidence for the major protection by this collapse. It protects the upstream xylem. Okay, so this is model world. What happens in the real world? Well, this is much harder because this is a very dynamic system. So I want to show you the results of an experiment. I'm just going to show the one we did on red oak, so staying with the same system. And our idea was we want to know what happens under fast dehydration, and we can characterize how embolism happens in the xylem from 0 embolism up to fully cavitated as water potentials become more negative. And we can do this well during a slow dehydration. This is typically what we do with the optical method, or really any hydraulic method. We tend to work in slow dehydration. We like to have things in equilibrium so we know what we're doing. But we wondered what would happen if along this curve, if there was evidence of protection, if you had a fast dehydration, would the plant, if this collapsing mechanism were efficacious, would you in fact not see embolisms? Would you get a divergence from this line that you have during slow dehydration? And the real problem was how to do this, and particularly how to do it in red oak, which is the species we knew the most about the collapse. And the problem was we couldn't generate the sorts of water potentials in the lab that we needed in a transpiring branch that we needed to affect either collapse or cavitation. So down in this range, if we had a branch and we supplied it with water, there wasn't enough. We needed the whole plant to generate these negative water potentials. So we did an experiment that we sort of colloquially known as, we called it the cut and run experiment, because we would take branches, transpiring branches from the top of a tree, and we knew what was going on. We would take it off, and then we would move it as quickly as we could to a microscope so that we could then use the optical technique to look for cavitation in the veins and to measure what was happening with embolism in the veins, and also freeze leaves, measure water potential, but also freeze leaves for collapse. So here are the data from those experiments. They were difficult to do. And the red curves just show the slow dehydration. They're from an optical method. And the open circles show the water potentials of leaves that were experiencing this. So these that were cut from the branch, and then we were exposed to a heating to hot air of an amount that we had predetermined should double transpiration. And then we've had some leaves that we froze for looking at the hydraulic index. And you can see there was, they're not up at the, these guys are at the 0.78. That's like a hydrated leaf. And these leaves that fall off of the slow dehydration curve all showed significant amounts of collapse, and they showed significantly less or substantially less xylem embolism than the slow dehydration curve. So at the predicted, from slow dehydration at these water potentials, you would predict a lot more cavitation than was observed. So this is sort of our evidence for a protective effect of collapse on during rapid excursions in water potential. And that's sort of where the question is, I think. And I wouldn't say this is a rock solid finding at the moment. We know the minor veins collapse, and the question is it a design outcome, an evolutionary spandrel of having to both be strong enough to sustain tensions during when the stomata are open and porous enough to let the water get out to the mesophyll, or is it in fact there's some component of this that is functioning as a protective valve, as a rapid and reversible means of buffering the upstream water potentials of the xylem, which is irreversibly damaged by cavitation. And I think this is a really interesting area because I'm really interested in thinking about design constraints and about trade-offs. And I think to go forward, we're going to really need more research under, real world. So non-steady state conditions that incorporates what stomata are doing, what capacitance is happening, and how resistances, transport properties are varying under these conditions. Now, these are really difficult experiments to do, and we'll need new measurement tools that work under unsteady conditions. So this is kind of like the next frontier, I think, in plant hydraulics. But I do think it's exciting. I think this is as we get out and work in plants in nature, and we realize that, the conditions inside a gas exchange cuvette or in a, as we equilibrate things for hydraulic measurements, are incredibly informative, but they are not the be all end all of understanding plant hydraulics. But the payoff of doing this hard work will be insights into how plants maintain their functioning in a, highly dynamic and ever changing world. So I'm going to now say, thank you to you guys for listening. I'm going to show the current members of my group, and some have gone on. There's Kate Johnson, who we had such a wonderful visit when she was here. And many people who contributed to this work are not shown in this picture because some of this work they've moved on to their own labs and their own futures. But anyway, thank you very much. I'd be very happy to take questions. And I think I will stop share, and I can always show a picture if need be. Oh, no.
Fantastic, Missy.
Thank you, guys.
Thanks for that fragile question. That was some beautiful images of leaves in a very fragile state. I love it. If you've got questions, stick your hand up. We've got one that's in the chat, or that came up from Elaine Mateos that just said was just asking about whether you're seeing evidence of collapse in the major veins or the midrib, or
So we've never seen, oh, hi, Brenda. Oh, that's fun. We've never seen collapse in a major vein, but, Hervé's paper in those pine needles, he saw it in the axial pine needles. And so, but we never saw that in Taxus. But the question says, can xylem conduits in leaf medium? I'm not sure what leaf or major, maybe, or is it only in the minor veins? We looked primarily in the minor veins. So I don't know in sort of second order to third order. I couldn't say. We were always a little worried. I think we're more worried than we needed to be initially, that the freezing process might itself induce cavitation. We just never saw cavitation in our leaves. We could measure it. The petioles appeared to be more sensitive a little bit than the veins, but when we really didn't see much cavitation in our leaves. And probably we should have taken our leaves out to more negative water potentials till we finally pushed them to cavitate.
Yeah, cool. So I think I'm not very good at navigating hands, but if you have a question, I saw hands and then they disappeared. But if you've got a question, put your hand up. Otherwise, I'll just keep on with these. There's a question in the chat. Do you want to read that, Missy? Can you see it?
Yeah, so collapse in veins of annual species and crops. Yes, we have seen it in a number of. So we've not done it's very time consuming to do all the cryo across a long thing. So we've done more spot measurements in a variety of crops, some woody, some herbaceous, and we've seen them. The only things we haven't seen collapse in was bamboo. But again, I can't say it's a totally exhaustive survey. So yeah. And then I've got another question about transmembrane flow through aquaporins activation in the model. Could it have changed the response? Couldn't it have changed? Yes, that is a possibility that there could be, that's absolutely right, that there could be changes in the mesophyll that would affect the rehydration. It could be true. Let me think about that. Yeah.
It's pretty fast reversible, isn't it? You would have thought aquaporins would be less reversible.
Yeah, I mean the yeah, I don't have I think it's a really difficult but important problem to separate the hydraulic contribution of the flow through the xylem, the flow through the bundle sheath, and then the flow through the mesophyll. And we and other groups are working to try to do that. And but we've not been able, I couldn't say anything specifically about red oak in that sense. So.
I see Greg's got his hand up, keen to ask a question.
Sorry, Missy. No, can I ask you about bundle sheaths, sort of lignified bundle sheaths? Do you know if they play this game as well? And if so, I might share can I share my screen? Is that going to work?
That'd be awesome.
I reckon you could, Greg.
I'll have a go. And can you see that?
Yep.
Yeah.
Okay, so what thing on the left, these are both proteaceae, which, like I play with. The thing on the left is a protea, and what you've got here is you've got these bundle sheath cells which are lignified, and so there's the vein there. This is a cross section. I apologize for the quality of my image, and this is a paradermal of another proteaceae, where you can see this is the terminal ends of the vein, and you can see that it's just incredibly lignified. So any idea what the difference there might be and what might cause those kinds of differences?
So one of the things that I think the mechanics of the bundle sheath, I think you're absolutely right on that it's really important. And, by just mechanics of, once a tube starts to buckle, I mean, if it were unconstrained in any way, they should buckle quickly. The fact that these guys sort of seem to lose, they don't collapse completely. They deform slowly. And I think it's because they must, they have to deform, they're attached to other cells, and then particularly to the bundle sheath. So thereby, I think the mechanics, the deformation of the bundle sheath, which we see in the cryo-SEMs, is so incredibly stretched. And I think that they're holding back this deformation, or at least slowing it. And that's why it's occurring over a range of water potentials. So by that argument, it could be that the lignified, because I presume they're lignified, but they're living cells, so not collapsing, but they're going to provide sort of like reinforcement to the bundle itself. I don't know what sort of water potentials this plant might experience, Greg, but maybe the bundle sheath is adding, the lignification is adding strength to the xylem, at the same time potentially retarding water permeability.
Well, those two species, the one on the left, which has got pretty feeble sort of bundle sheath things, and sometimes these cells can be really big. It's actually probably from a drier climate than the one on the right. So the one on the left's a protea, so South African sort of Cape Mediterranean climate thing, and the one on the right's Bellandina, sort of, which is Tasmanian subalpine kind of thing. So that's why I'm really confused.
Yeah, it's true. I mean, you'd have to know what sort of tensions they respond to, but they operate at. But yeah. No, it's cool. It's a beautiful picture. I love the one on the right. I thought it was an algae, to be honest. Like a fucus.
Good one, Greggles. I see I see Brendan's patiently waiting to have his hand up.
Hey, how's it going, Missy? Great talk.
Good.
Thanks for that. Yeah, I was just thinking a bit about some of the work that had come out of Lauren Sachs' group and Christine Scafoni. It's a few years back now, but they were also showing very large declines in K-leaf before they had any signs of cavitation, and they had complementary measurements from micro-CT and evaporative flux technique. I remember thinking at the time, I was vaguely skeptical about that big decline in K-leaf, how affected it was just by the stomata in the way that things are measured with the evaporative flux technique. I think they ended up attributing a lot of that early loss of K-leaf to aquaporin activity. I could be misremembering that, but I don't remember much discussion of conduit collapse in those papers or any.
No, I think that, so most of, as I understand, most of the declines that have been reported happens at pretty positive water potentials. You know, that's the majority. I mean, they see a decline throughout, but a lot of it is happening way before any reports of conduit deformation than I've seen. They did talk about changes in cell to cell contact that could occur upon drying or desiccation. So I think that, you know, methodologically, there's that measurement shows that there's a lot of loss at a very high water potential that I think doesn't, I believe the conduits have to be open or, uncollapsed for transpiration to occur. So I would think that those would be tensions that wouldn't cause, really, you would cause any problem. So I think it has to be a different mechanism.
Yeah. I also wonder, if there's any, you talked about kind of, the methodology that you'd need to really nut this out experimentally. I wonder if those imaging techniques like micro-CT might offer opportunities to do that. I guess it depends on how quickly you can image and what conditions you can put leaves and plants under inside the cabinets or the hutches, but you can certainly get the resolution to see those kinds of things.
Absolutely. It's just hard to get the tensions and the transpiration, the dynamics to happen. You can get one or the other, you're just not getting both. It's those trade-offs again that are killing us.
Yeah. And I missed, you had that beautiful image which showed the terminal tracheids in a leaf. What was the technique? Was that fluorescence confocal?
It's a confocal. Yeah, it was just confocal. I don't know what stain was on there. It was or what the wavelength was. It was more done for aesthetics, but it does show the structure.
Yeah.
Very pretty.
I wonder if there are any we've heard some old fogies talking. I just wonder if there's any ECRs who've got early career people who've got any burning questions, because it would be nice to see a few hands up from, people in my lab, for example. Or I'm sure there are some questions, but I've got
hi, Tim.
Ibrahim.
Just got a question. Hi, Missy. I was just wondering, does that mechanism of conduit deformation occurs on a daily basis during daytime transpiration?
So that's a great question. I don't know. So I have more questions than answers. I don't know how much, transpiration is bouncing around, and I don't know.
Because you've showed that the deformation occurs in parallel with the stomata closure, and we know that stomata close on a daily basis under high VPD. So I was wondering if those two processes occurs simultaneously.
I think I think it's entirely possible that the conduits, in a rapidly transpiring leaf are always sort of just right on the verge, of changing their conductivity. I think that's possible, but I can't. That's just my imagining that's I don't have a measurement that suggests that, it is the right range of water potentials that plants can operate at, before the it's right before the stomata close. So it is possible, but
Yeah, because that mechanism sounds a little bit dangerous to the leaf function itself, because if because you showed that graph where leaf temperature changes quite drastically within minutes, and so if you increase the VPD, especially in angiosperms, and that they show like a wrong way response, and if K-leaf declines because of deformation during this transient drop in leaf water potential, it could basically amplify the wrong way response and further decrease the leaf water potential, and it's just like a perfect ingredient for.
You raise a very good point, Ibrahim. And one thing I didn't point out. So one of the nice things about the fact that, well, nice, or maybe a feature, that these are not like, they're not like a digital on off. The closure, there's still some water coming through the xylem. It's just being choked down. And therefore it doesn't leave the water potentials don't plummet, because there's, you saw in my model simulation, our model simulation, there was still some water coming through the xylem. It's just the potentials were reduced because the resistance had gone up. And I think that's this might be something about the geometry, the way the cells interact, that the collapse doesn't go from open to close, on a dime, but chokes down. So in that sense, I think it is a little bit more robust. But you raise an excellent point that, yeah, if you started doing that, you could get yourself into a runaway.
Okay, thank you.
Yeah, good question. All right, well, we've come to 11 o'clock, and it's pretty late there in the US now. So
Dark here now.
really, thanks so much, Missy, for that great talk and that nice discussion at the end. We'll let you go and have some dinner and relax after what's probably a pretty long day. Yeah, so thanks from everyone here, and I'll talk to you very soon.
In touch. Nice to see Brendan, Greg, and everyone else. And reach out by email. I'm always available. Happy to talk.
Awesome.
Thanks, everyone.
All right. Thanks, Missy.